DNA.
DNA replication by Troy Zarumba, Morgan Locker, and Jake Zara. Helicase is an enzyme that untwists the double helix at the replication fork, separating the two old strands.
When helicase unwinds the DNA at the origin of replication, it creates a bubble and a replication fork, which is a Y-shaped region where the new strands of DNA are elongated. The single-stranded binding protein holds the DNA apart while they serve as templates for the synthesis of new complementary strands.
An enzyme called primase joins RNA nucleotides to make the primer.
This is the lagging strand of DNA. Because DNA polymerase can only work 5 to 3, on the lagging strand, it must keep jumping back in order to keep moving in that direction. Because DNA polymerase must keep jumping back, it is unable to connect the new DNA being made to the previous primer that it was connected to, causing Okazaki fragments.
After DNA polymerase does its job, another enzyme called DNA ligase joins the Okazaki fragments into a single DNA strand.
Thus you have the corresponding 5 to 3 pattern. Because the complementary strand of DNA on the leading strand is 5 to 3, the DNA polymerase can just race through and continuously make the complementary DNA strand. DNA proofreading.
There are two types of repairs, mismatch repair and excision repair. During mismatch repair, DNA polymerase fixes mistakes that are made when DNA is copied. DNA polymerase itself carries out the mismatch repair. It proofreads the strands and then fixes the nucleotides against its template as soon as it is added to the strand.
Excision repair is the result of a DNA cutting enzyme, nuclease, cutting out faulty nucleotides and replacing them with the right
After
ones.
the faulty nucleotides are cut out due to the help of nuclease, DNA polymerase comes back in and it places the correct DNA nucleotides in their place. After this, ligase comes up and seals the final DNA strand.
Eukaryotic chromosomal DNA molecules have special nucleotide sequences called telomeres at their ends. Telomeres do not contain genes. Instead, the DNA consists of multiple repetitions of one short nucleotide sequence. Telomerase is a special enzyme that catalyzes the lengthening of telomeres. As you get older, your DNA gets shorter and shorter because you run out of telomerase. That is why all cells must die. Thanks for watching.